So this is a second learning sequence in this block where we are going to discuss inferential statistics, how to get a knowledge about your population if you know something about your sample. I'll start with introduction, where I'm going to repeat a couple of things, quite important things about relationship between samples and populations. Then we will discuss sampling distributions, we will discuss confidence intervals, hypothesis testing, and most of the time it will be based on the one particular parameter. It's an average value. Okay. So I'll remind you quickly what population and samples are. Population is a set of all individuals of interest. So if you, for example, would like to know chloride content in the whole river, population will be every single drop of water in this river. And then since the population most of the time is huge, you cannot actually work with the population. So you take a small portion of the population, which is a sample. And then the individual measurements that you do for your sample, or individual objects, individual physical samples, your statistical sample, are called individuals or just objects. Okay, that's the data set we are going to work with. If you remember our short class on numerical methods in our analytical chemistry on the third semester, you'll find out that the example is very similar to this one. The idea is following. We have two rivers, Vada and Riba, and we would like to know if they are contaminated. Let's say we would like to know the chloride content in the rivers, and we know that if chloride content in average is above 146 milligrams per liter, then the river is contaminated. Okay, and then we have two rivers, Vada and Riba. Since we cannot take every single drop of water from these rivers, we took 10 glasses of water and estimated how much of chloride is in each glass. So we ended up with 20 values, 10 values, concentration of chloride in 10 glasses of water for river, and the same for Vada river. Okay, so that's the data set. Here are the questions we are going to answer in this learning sequence. We need to find out how to estimate average chloride content in the whole river based on these 10 glasses of water. We would like to learn how to compare the average values with a certain value that we pretend to know. For example, how likely average chloride content in the river does not exceed, for example, a certain value, in this case 146 milligrams per liter. And also we would like to know how to compare different populations and different rivers. For example, can we say that one river is more contaminated than the other river? But the answer to this question we will discuss in the next learning sequence, where we will learn how to compare the groups. Okay. So these are statistics calculated for samples taken from the rivers. And I have checked the values, and they seem to be taken from normally distributed population, so I decided to use parameters of normal distribution as statistics. So the average and standard deviation you see on this slide, you can see that the standard deviation is about the same for the samples, but the average values are quite different. We can see that it's 152 milligrams per liter for Vada and 139 milligrams per liter for Riba. So what can we say from these values? Can we say that indeed, the rivers are different? Can we say that, for example, one river is more contaminated than the other? Can we say that, for example, Vada is contaminated and Riba is not contaminated, assuming that contamination border is 146 milligrams per liter? So that's what we are going to discuss in this lecture. First of all, I'll remind you that sample is always an approximation of the population. So when you see average value for a sample, it doesn't mean that average value for the population will be the same. Moreover, it will definitely not be the same. Okay, so we know that in real life it's possible to take a sample and say something about the population. We discussed that in the last learning sequence. And we know that it's very important that samples should be random and representative, right? So only in this case inferential statistics will work. So we assume that our sample is indeed random and representative. And in this case, we have to use a special set of methods which are called inferential statistics. And you already know that inferential statistics is based on calculation of different intervals and hypothesis testing. So my goal in this learning sequence is to remind you the main things about confidence intervals and hypothesis testing. Okay. You remember this actually picture. So the statistical analysis usually consists of 3 steps. The first step is sampling, when you take a sample, and it's important that your sample is random and representative. Then the second step is descriptive analysis, when you calculate different statistics and use different plots to describe your sample. And finally, the third step is inference, is when you use knowledge about your sample to get a knowledge about your population. And that's what we are going to discuss in this learning sequence. First of all, a problem formulation. Why we cannot just use statistics of our sample and say is that parameters of population are the same. Let me show you a simple example. Let's say our population is quite tiny. It consists of 7 students. So I just denoted them A, B, C, D, E, F, G. And then we have IQ value for every student. And this is our population. So if I calculate average and standard deviation, we will have a certain parameters. In this case, the average IQ is 106.7. Okay, so they are fixed. But every time we take a sample from this population, the statistics will be different. First of all, I took a random sample, 3 persons out of 7. It's a huge sample. It's almost 50% of the population. But you can see that the statistics are not the same as the parameters of the population. Then if I repeat it again, if I repeat it again, if I repeat it again, even if I take a sample a little bit bigger, every time I take a new sample from the same population, even though the sample is big enough, the parameters, the statistics of the sample vary. So we do not have the same statistics. Sometimes it's pretty close to the parameter. Sometimes it's not. It's pretty far from the parameter. The problem is usually we don't know the parameter, so we have no idea how far the statistic of our sample is from the parameter of population. Okay, so that's just another example. In this case, I use a simulated example. I did it in MATLAB, and let's say we have a huge city, like 1 million inhabitants, and we would like to know average height of the persons in the city, adult persons in the city. So the first plot shows a first sample. It's a 20 persons randomly taken from the whole population, and you can see statistics, the average and standard deviation. Then I took one more sample, one more sample, one more sample. So if you look at these 4 samples, you can see that even though they were taken from the same population, they have the same size, they were taken randomly, perfectly randomly, you can see that the samples are quite different and the statistics are different as well. Okay. Why we have this difference? Because of the sampling error, because sample is not the same as the population. Sample is always different, and this difference is called a sampling error. I can give you an intuition what sampling error is using a very trivial example. Let's say this is our population, it's a curve. Then you take a sample of size, let's say 3, so this is your sample, okay? So these 3 points represent the whole population. So actually, if you make an approximation of the population, you will think that the population looks like this dashed lines. But of course, it's not the same as the whole population, right? You can see a clear difference. And this difference between the shapes of the sample and the shape of the population is a sampling error. And of course, if you calculate different statistics of your sample, they will not be the same as the parameter of population. And we know that if we take a bigger sample, for example, we say 5 points, 5 random points, then you can see that actually the sample will be closer to the population. The shape of the sample will resemble better the population shape, right? But still there is a difference. There is only one case where the sampling error is 0, when the sample is the same as the population. Usually it's called census in statistics. So every time you take a sample, you have the sampling error, meaning your sample will be slightly different from the population, okay? So what it means? It means that you cannot estimate population parameters from the sample. It's impossible because sample is not the same as the population. But it's not possible to do it precisely. We still can do something in this case. So let's discuss that.